Alrighty, hello everyone, and welcome to today's session. Today, we're going to be looking at the origin of life and how life started on the Earth from the scientific perspective in terms of between the Big Bang Theory and evolution. How did the life actually start in the middle in the first place? And so the prevailing theory is that there's the so-called primordial soup, and that there was just the right chemicals in the right way. They came together. That forms the first life form, and from there it evolved and adapted to its environment. And here we are, lizards, monkeys, and man. All right, so first we'll talk about why this stuff's important. Why do we care about ancient goop? Then we'll talk about the things you need to have life in the first place. So we'll talk about the Lego bits to make up protein, how proteins come together, because proteins basically do all the jobs of all the things in our body, very important. How do we store the information about how to build these proteins? How do we keep it in one place in a cell? And then how do those cells replicate and continue to make new cells and new life? So we've kept searching for the origin of life for a long time. And ever since the dawn of mankind, I suppose we've always been wondering, or well, probably not since the dawn, but for a long time, we've been wondering how we got here. And so we all have this innate desire to understand our purpose for existing. So even if it doesn't keep you awake at night time, kind of thinking and pondering these things, it has big ramifications after we die, of course, but it affects us right in the here and now. Because if you believe that you were fearfully and fearfully and wonderfully made, custom created by a loving creator who has a plan for your life, you're going to live your life differently in terms of going about that plan and using those skills. Whereas if you think you're just a random accident, you're probably just going to be here and have as much fun as you can before you kick the bucket and become worm food and nothing happens at the end. So it has ramifications for what happens after you die, but how you live your life today. And so that's why this stuff is important. That's why we care about ancient chemicals that may or may not have created life. All right, so let's start with the basic building blocks of things that do stuff in our bodies. And so that protein's important. We all need to eat protein. That's why it's a key nutritional ingredient. But proteins do jobs in our body. They do things, they do chemical reactions, they form structures and shapes. If there's a job, a protein generally does it. And so when we have drugs that help us to combat diseases, it's generally a chemical that's interacting with the protein. That's how we get that effect that's beneficial. And so these proteins are made of little Lego bits called amino acids. So they're just little Lego blocks to make it up. And they help those proteins to come together. So at the bottom here, I've got a string of a number of amino acids all connected together. So alanine, leucine, isoleucine, and so on and so forth. So they all join up in a nice big string like this to form a protein. Each of these little guys is one amino acid forming a chain in a protein. And so we have that all chain comes together. Each of these little guys have their likes and dislikes, and so they'll arrange themselves to be close to the ones that they do like, away from the ones that they don't like. And as that happens, it forms this unique and perfect structural shape, which is the protein. And each protein has a specific job, and its job is designed around its shape, that specific shape. So you can think of the protein and the thing it does the job with as like a lock and a key. They have that perfect fit. And so a drug might be a key that fits in the lock of a protein to make it do a change, or the protein might act on a lock inside the body. It might be a chemical inside the body to do the thing it needs to do. So very important stuff. And so obviously we need these things for life to have happened, because you can't have life without things that do stuff, because then there's no life happening. And so Miller did an experiment in 1953. And he wanted to see if these protein Lego bits, these amino acids, could just pop into existence under the right conditions. And so he set up an experiment to mimic what scientists' best guess was the early Earth. After the Big Bang event and after things had settled down a little bit and the Earth had formed, what were those conditions like? And could we just produce these just by happening, just spontaneously? And so he set up his experiment like this. And so he had a bunch of boiling water, made things really hot, and so that had turned to steam and go up through the pipe. And he'd introduced a whole bunch of other gases which they think were present at that time on Earth based on what they find in rocks and that sort of thing, gases trapped in rocks. And so all those gases mixed together, high temperature with water, zap it a bit, because apparently they think the early Earth was very electrostatically charged, lightning bolts, whatever. And so there's that little bit of spark to get things happening. And so as you get this heat, all these ingredients and a bit of spark, do they come together and do they form some amino acids down here? And so he let this run for a week, and he did it at 800 degrees Celsius, which is pretty warm. I prefer a nice 24 degrees. But basically at the end of the week, they found a number of amino acids down here and concluded that huzzah, amino acids can just spontaneously form in these early Earth conditions. And so that's really cool. That's some nice evidence to show that that's a possibility. But I do have little queries and issues with that. The first one is 800 degrees realistic? I don't know enough about early Earth science, geology, that kind of stuff. Maybe it was that hot, maybe it wasn't. But in theory, after Big Bang, things are still cooling down. Different distances from sun, atmosphere, all that sort of stuff can affect temperature. So maybe it was quite warm. But even if it was that warm and we did see these amino acids in those conditions, amino acids have a habit of degrading at high temperatures. So yes, you can form them, but they don't hang around for long. They break down. Heat gives things a lot of energy. They move, they jiggle, and jiggly things just have a habit of falling apart. And so they degrade at those high temperatures. And so next is, and the amino acids that formed would have a 50-50 chirality. So chirality is a fancy science word for mirror image. And so basically there's an amino acid that might kink this way, and then you might have another one that's totally identical in the bits that make it up, except it just kinks that way instead. And so that mirror image version of each other means there'd be a 50-50 split of each, if they were just created by chance. But our body only ever uses one of the kinks. This one isn't recognized or used by the body at all. And so all of them are just the one version of mirror imageness. And so even if all these chemicals were created, only half of them would be used and, give rise to what we see in living organisms today. And then lastly, they made amino acids, but they didn't make many amino acids. And so I've linked to the Wikipedia article, and I've pulled a table from that as well. Wikipedia, great source. And so you can see the experiment here. They produced a whole bunch of amino acids: glycine, alanines, aspartic acid, and butyric acid. So cool, they made some amino acids. And then in recent years, they've kind of done other experiments too. So they did that's volcanic spark discharge and a hydrogen sulfide rich spark discharge. So they use different gases, different conditions, and boom, they've made a whole bunch of other amino acids, as you can see here. And so that's interesting. They kind of built on that. They've shown that more amino acids could be formed. But the really important column is, are they this one here? Do they actually give rise to life? And so the greens here are saying, yes, these guys are actually life-giving amino acids required for life. So humans, we have 20 amino acids that our biology is built around, and that forms all the different proteins that we use. But you can see here that, they made a lot of amino acids, but like a lot of them aren't actually used in bodies and life at all. They're not actually helpful. And as we go over the page, there's even more, a lot of amino acids. These guys down here, these are the very familiar ones that you might have seen in a science textbook, because they're the ones that help us to live. These are all the greens saying that, yes, we need those to live, but well, these experiments are not good at making these. And so, yes, there's evidence we've shown some amino acids can pop into existence spontaneously at extreme temperatures, but it is, a minority of the amino acids that would be required for life. All right, so we've looked at the building blocks of, do we even have the Lego to build a protein? What's required to actually make a protein happen? And so on the right here, I've got a picture of a ribosome. And so this is a protein. And so this is a diagram representing all the chemicals that are going into making it up. And so this is a chain of 7,459 amino acids together. And each one of those 7,459 amino acids has its friends and its enemies, and it will arrange itself in this perfect shape just spontaneously, because these guys want to be together, these guys want to be far apart. And so you can see it forms this shape, and this shape is what allows it to do its job. What does the ribosome do? It helps to build proteins and stuff. And so that's that active site in the middle is going to be where, the lock might go, if it's, where the key might go, if you think of it as the lock. That perfect shape allows it to do its job. And so for these amino acids to build a protein, they have to be connected. And so amino acids have an amino end and an acid end, and that's the kind of backbone that connects them all together. And so for those to connect, that requires a chemical reaction, and that chemical reaction requires water. And there's not a chemical reaction that happens naturally. They don't want to do it. You kind of have to force that chemical reaction to happen. And so we use catalysts to force that chemical reaction to happen quicker. And what is that catalyst? It's a protein. So to make a protein, you need protein. And so we get the chicken and the egg problem. What came first? How do you have one without the other? How does that work? And so evolutionists have said, well, okay, maybe obviously it wasn't this fully formed protein that did it first. Maybe there are some simple forms of this. It was slow and happened over time, and that kind of thing. Arguing as to how those proteins may have come together. But you can see this massive complexity now, and just that specificness. And it's really hard to convey how unlikely these reactions are to occur without a catalyst of some sort. You have to really force.
Actually, it's pretty easy, because a ribosome is a machine that is present in our DNA.
Oops, someone trying to talk there. Sorry.
Yeah, me. Sorry, Tim.
What's up?
So ribosome. Okay. So you've heard of DNA.
Yeah.
Within DNA is the machines that your DNA builds to build DNA.
Yeah.
Ribosome acid. RNA, ribosome.
Yeah.
So that's what you're showing me for, is ribosome.
Yeah.
So that actually creates, the information to create the ribosome that you just saw then is in our DNA, which then is the machine that actually makes sure everything's in the right place in our DNA.
Yeah, exactly. We're going to get to that in a moment as well. It's like layers upon layers. It's kind of bonkers. It's how it all works together.
It's actually not just a random, a really well designed thing. It's actually a machinery.
Yeah, absolutely. Little machinery. Yeah. All right. And so speaking of machines, we need blueprints. That's a nice segue to the information storage, for the blueprints to make this stuff happen. So we've talked a little bit about DNA in the past and how that works. So probably familiar with the structure of DNA, how it looks like this twisted ladder shape. But if you untwist the DNA, it's a ladder basically, and the rungs of the ladder are these little letters, G's and C's and A's and T's. And so these are just little chemicals that connect to each other, and each chemical basically tells the body to use a certain amino acid. And by reading these in sequence, it'll tell you to use this amino acid, then that one, and it'll tell you how to make that protein and what that looks like. And so this is how our body stores that information to make the proteins, so that the proteins can do the stuff that our body needs to make happen. And so in of itself, this is just a chemical. It's useless. It's just a nucleic acid. And that's why for a long time, scientists were just ignoring those weird nucleic acids. We don't care what they are. And then in the 50s, someone was like, hey, they're probably really important. Maybe they're the entire basis for genetics. And since then, molecular biology has kind of grown based on that. And we can do that really cool stuff that we do today. And so this is just information. So it needs to be read. We need to read that information. Again, a protein does that reading. And then that arranges for the amino acids to make a protein, reading that information from this DNA blueprint. And so that's cool. We'll look at more about how that happens in a moment. But I just want to highlight here that this is a huge energy investment for a cell as well, because not the rung, but I don't know, the up and down part of the ladder, whatever you call that, that's a sugar phosphate backbone. And so half of this is basically sugar. That's a lot of energy for a cell to invest, because a human body has, about 70 trillion cells. And each of those 70 trillion cells have the entire DNA of 3 billion letters back to back. So that's a whole lot of sugar just to store information. And so we'll talk about why that energy is important in a moment as well. But another cool fact is that DNA is the only example of information code that was not made by humans. We've made Morse code, we've made music, but this is the only code found in nature. And so a code implies intelligence. And so for me, I'm like, duh, something intelligent had to have come up with this coding system. Alrighty, so we've talked about why DNA is important. We need that information to build the proteins that makes life. So how do we get from a blob in primordial soup to a human which has all this complex DNA, 3 billion letters back to back to back? And scientists theorize that basically, you might have had a simple string of DNA that's just come together in the beginning, and maybe these little blocks represent little genes in that DNA. Each of those genes makes a protein which does things. But over time, as cells grew, as cells divide, they actually need to copy the DNA, have 2 entire copies of the DNA. So when your 2 cells split, each of those cells then has its own complete copy of the DNA. And so the theory is maybe that a cell copied the DNA, but something went wrong, it didn't split, so it had twice as much DNA inside it as it needed. But for some reason, that DNA just hung around. And over time, it does its evolutionary mutation-y thingy, and those 2 copies of the DNA will actually look different over time because they mutate in different ways. And that's indicated by the X here. And so over time, they might look different. And then maybe later on, it accidentally doubled again. So I've got even bigger, even more DNA. But over time, you've got more strands of DNA to mutate in different ways. And so that's kind of the idea of how we get from little sludge to the complexity, all the genes and all the DNA that we see in complex organisms today. But like I mentioned before, this is where that energy and nutrient requirement comes in. Okay, cool. It could happen that we double that DNA. Okay, yeah, it might mutate over time and see these different genes. But that is a huge amount of energy, is a huge burden on that cell to be, pumping out all that sugar and all that phosphate to have these additional nucleic acids, which actually aren't required. And the smaller an organism is, the bigger a deal energy is. So I've worked in a lab with E. coli, just a little bacteria, and we actually put genes into it to get it to make proteins for us, like a little protein factory, basically hack it and steal its proteins and kill it and take it all. But basically, it's really hard to get those organisms to take in DNA because they don't want it. It's a whole bunch of energy and they're lazy. They don't want to do that. And so they'll spit it out, or they'll basically choose death instead of taking it on. And so you actually have to put antibiotic resistance in it and then put it in antibiotics. So you kill all the ones that don't take it on, you kill all the lazy ones. And so it doesn't make a whole lot of sense to me that organisms would take on this huge bunch of random DNA that's actually not needed and would take, thousands or millions of years or whatever before it would mutate into something useful. That's crazy to me. Even if they didn't spit it out or get rid of that DNA, they would be have this huge energy requirement that would slow them down to the point where they would be outcompeted by other organisms essentially and cause them to die out. All right, so we'll come back to DNA in a moment and how they actually make the proteins. But first, we've talked about the building blocks, how they make proteins, how the proteins come together. We've talked about the DNA blueprints to make those proteins. And so, yeah, you got some information. Yeah, you got some proteins which do stuff. Cool. To be life, you need to be a cell. To be a cell, you need to contain things like a prison cell. That's how they got their original names. They look like cells. And so we need this cell membrane to keep stuff in it. And so this little graphic on the right is a cell membrane. And a cell membrane is made up of 2 layers of fatty lipids. These are fats, basically. And so you might have heard of saturated fats in your food. They're ones that have a nice, perfect zigzag. The unsaturated fats, they have a little zigzag and a kink in it. And that unsaturation causes them to be a little bit bendy like this guy. But basically, they have this interesting features where the guys in the middle, these are all just big strings of fat. And big strings of fat, oil and water, fats and water don't mix. And so these fats will hang together. They'll all hide together, and all the water will hang around upside here and down in there. So the fat will actually kind of push the water out, and all the fats will hang together. And then the top end, where the red dots are, these are actually water-loving parts of the fatty acid. And so these guys are happy to be chilling on the outside with the water, and these guys are happy to be down here chilling on the inside with the water. And in the middle, all the fats can hang out together too. And so when you put these chemicals in water, you put this fat in water, they will just spontaneously arrange themselves because they want to exclude the water from the middle part where the fatty tails are. And the red heads, they're happy to hang out with the water too. And so this kind of makes sense. Yeah, this we can see this happening. It just happens. It just pops together just because of the chemical makeup. So it'd be pretty easy for some fats to come together in a way, where it would surround some proteins or surround some DNA to make, the first cell. So that's kind of an easy cell in terms of this type of thing. But with that said, life is not that simple. Yeah, we can find a capsule for stuff, but even a prison cell needs things to go in and come out. You need to be able to remove waste. You need to be able to put food in. And so a cell membrane actually looks a bit more like this. We've got the red heads on the top and the bottom. We've got the yellow tails in the middle, but we've got all these other weird shapes in here. And these are all proteins which have a job, proteins which do stuff. And so this is a protein tunnel, which allows things to go in and out. And a bunch of these guys are for signaling. So glycoprotein might touch another cell and, tell it what it needs to do. Or, a signal might come along, maybe insulin comes along, it might stick onto one of these proteins, and the protein would then send a signal inside the cell, and the cell would then have a response in regards to that insulin and pack the sugar away and store it for later, that sort of thing. And so a membrane's great at keeping things in and out, but we actually do need some things to go in and out. We need amino acids to come in and out so proteins can be made. We need sugars so things are fed. We need ions and salts just so those electrolyte balances are right. And we need water because, water is important. We're 70% water. Things need to drink. Water is part of reactions too. They need to move through the water. So we actually need a whole bunch of proteins in this membrane for stuff to happen. So yeah, a cell could come together, but where did all these specialized proteins come from? How did the important stuff get in and out in the beginning? How did all this stuff just kind of fall together? Knowing that each one of these things is a very specific thing made of very specific parts to do a very specific job. All right, so we've talked about all the bits, how they come together. This is what Tim was alluding to before in terms of replication. And so this is a diagram of how DNA actually gets copied in our body. So when a cell's getting ready to split or divide, it needs to make a whole additional set of DNA so that when those cells split, they can have more DNA, and this is how it happens. As you can see, there are proteins everywhere. So we've got topoisomerase, which unwinds the ladder, so it's nice and straight, so you can actually do stuff with it. Helicase here actually splits, the steps of the ladder apart, so we can access each of the individual letters and read them. And then DNA polymerase comes here, and it will actually take one side of the steps of the ladder, and it will fill them in with the other side to complete the ladder. And so the ladder gets split in half, and then each of those halves gets made into a complete new ladder, essentially. And that's how the DNA gets copied. And so that kind of happens easily on this side, but the DNA proteins are actually copy and write in one direction only. And so this side's fine, but on the other side, it actually needs to do a bit and then go back and then do a bit and go back and do a bit and go back, because it only operates in one direction. And so we need an RNA primer to get those bits started. Then we need a DNA ligase to connect those bits together because they come in like little fragments. And then the DNA polymerase on this side to do all the copying. And then a DNA primase gets all of those steps ready to be written on. So you can see there's like what, 1, 2, 3, 4, 5, 6 different proteins required here. And then single strand binding proteins, which help facilitate that too. So we've got proteins going everywhere. Nucleic acids, all these bits with a high energy requirements. And so there's a lot going on just to copy some information from one cell to another. And so ask the question, how do we copy DNA in the first place if you need all this stuff just to make it happen? And if all that stuff requires a DNA blueprint in the first place to make happen? And that's just at the DNA copying level. There's the DNA storage as well. So we looked at the DNA here and what that looks like. It can't just float around because it's only 2 nanometers wide. And even just swooshing it in water because it's so long will just cause it to snap. If like it's like when you got Play-Doh and you stretch it out, it gets so long, any movement's going to snap it. But if it's in a blob, it's pretty strong. It's going to be okay. And so we need to actually wrap this DNA up to store it safely for next time. And so these little strings get wrapped around proteins again, histones, little beads, basically a string of beads. Then those beads get packed together, and then they form little ropes, and then they form bigger ropes. And they all kind of come coiling together, and then you get this big X shape, which you might have seen before.
Chromosome.
Yep, and this is a chromosome, that's right.
Yeah.
So all of our DNA is bunched up into a bunch of chromosomes to keep them nice and safe. And when it's time to come and do that DNA copying, that needs to be all unpacked and unraveled so that we can safely access it with all these proteins to do this process. And so in humans, this can actually write 50 letters per second. It's really quick. And in bacteria, it's a bit more rough and rugged, and they can do 1,000 per second. And so this is all just happening incredibly quickly, all this unfolding, all these proteins flying everywhere, and then all the ingredients, all the nucleic acids just coming in. And yeah, it's a really cool process. There's a lot going on. Very complicated in how it works on its own. All right, so that's the information level of replicating. Then there's also like, how do you get a parent and a child replication happening? So initial bacteria, that they just divide, they just split, they make clones of themselves basically. But these days we have more complex organisms, and so you might have a male and female. They might undergo sexual reproduction. That allows the genetic material to mix, which gives it an advantage in the environment instead of just cloning itself. Because if a clone isn't well adapted to environment, that whole species is going to die out because they're all the same essentially. And so logically, okay, I can understand the bacteria happening. They split, cool. They survive, okay, they're lucky, whatever. But the first male and female, how would that happen? Because the first male would have to be around at the same time as the first female. They would then have to be genetically and physically compatible and have children and not die. And, it seems incredibly unlikely that all those things would come together if it is so difficult for that thing to happen, if it takes millions of years to create, the genetic changes required to have one new organism feature in this way. And so I actually looked at how scientists theorize this happens. I didn't go to the literature, but I did find some cool like science news articles, some interesting names. And so there was an article about isogamy, called How We Evolved into Male and Female. I'm like, cool, okay, that'll explain how we evolved into male and female. And I read the article and it had no description whatsoever as to how this may have actually occurred. And then I read another one about how the sexes evolved, and then I read it and had no description as to how this may have actually occurred. And then I read another one called Male or Female First Sex Determining Genes Appeared in Mammals some 180 million years ago. And again, this had no description of how this may have actually occurred. And so these articles were great at saying, hey, this gene popped up at this time based on my evolutionary timeline, but very little in the way of, yeah, but how? Or they talk about individual genes and how they might mutate, but how does that come together to form something that's functional and viable and will outcompete others and has, a biological advantage in terms of these genders or whatever? There's very little in the way of that. They just looked at these genes and said, hey, it's cool that this thing happened, but very little in the way of how it may have actually happened. All right, so let's just recap on the practicalities of what's required to get from soup to a bacteria, or maybe like a male and female organism of some sort. And so we're talking chemicals forming amino acids spontaneously at the same place at the same time. Okay, so we're showing that, yep, that might happen. So they need to combine in a meaningful way. So not just amino acid here, amino acid there. They need to be together. They need to connect. They need to do things. They need to connect with reactions that rarely occur spontaneously in the conditions that we identified before, cause things to degrade very quickly. So we're seeing a very, very unlikely sequence of events happening. And even if you did get a fully formed and functioning protein, it then needs a way to reproduce itself. And over time, it needs to become encircled by a cell membrane. Yeah, that might happen the first time, but how do you get stuff in and out? From that, you need the blueprint to keep replicating. Where did that come into things? How did that stuff happen? How do we read it, not just have it, the information? And then how did that information, how did the amount of DNA grow without being supreme disadvantage in terms of the energy it requires? And then it needs to mutate in a way that was actually useful, even though that's incredibly likely. If you go changing letters in a Shakespeare play, you're unlikely to make it better, just changing things at random. And somehow over time, they became more complex, and then they moved from water onto land and didn't die immediately and kept breathing. Then there were some males and females that had to find each other at the same time and place, so genetically and physically compatible, and their offspring didn't die. And yeah, they weren't all wiped out by things. They have to repeat and do it all again for all these different other species. And so that's just like a series of very unlikely events on very unlikely events on very unlikely events. We've talked about the fact that, yes, some of these things could happen. That's valid. But let's have a look at the probabilities of some of this stuff. And so Fred Hoyle wrote a book called Evolution from Space, and he estimated that the odds of one cell appearing with the right enzymes for even the simplest life is 1 in 10 to the power of 40,000. And so that's the odds of like half of this page happening, 1 in 10 to the 40,000. And so winning the lotto, we generally accept as impossible. That's 10 to the 7. 10 to the 40,000 is not 39,000 times more likely, unlikely. It is 10, as a 10 followed by 40,000 zeros times more unlikely. And so, you know, I'm yet to win the lotto once. And so for that to happen, just to form one cell with one protein that could have just been gone because the wind blew the wrong way at one time is absolutely mind-boggling to me. And just to put those numbers into more perspective, the number of atoms in the entire universe is 10 to the 80. Every proton, no, not proton, neutron, every like hydrogen, helium, oxygen, and carbon in the entire universe is 10 to the 80. A teeny tiny number compared to 1 in 10 to the 40,000. So much to the extent that mathematicians basically say, yeah, 1 in 10 to the 50, not happening. Mathematically impossible, just about. And so for one cell to come into existence, bonkers, bonkers upon bonkers, unlikely upon likely. So it's been likened to the odds of just one enzyme, so not just the cell, but one enzyme within that cell is basically a solar system full of blind people solving a Rubik's Cube as each other at the same time. And so God or not, it's certainly a miracle that we are all here based on those probabilities. All right, so we touched on why the origin of life is important because where you come from and where you're going, you know, it factors into how you live your life every day with your attitude and obviously how you're going to spend your eternity as well. And so we looked at the processes that must have occurred for life to happen, looked at amino acids and how they might form spontaneously, how they can come together with great difficulty to build proteins, the information storage in DNA, the blueprints that you know, allow those proteins to be made again and again the same way, then how it's all stored together in a cell membrane, and then how can it keep copying it so we can do it for the next generation for next time. And then lastly, got some references for you guys to read on. So I've got that Wikipedia link about the Miller experiment. I've got those three links. I'll copy the link there so you can see those news articles for yourself. Then there's the last one that I haven't touched on, which has a general overview of life's origins from George Washington University. I just kept coming across it and it was really balanced. So that's well worth looking at. But yeah, as always, thanks for listening and I'll see you guys all next time.